My name is Grayson Huston. I'm a graduate student at the University of Maine, pursuing a degree in ecology and environmental science, and in the School of Biology and Ecology. With Maine-eDNA, I'm looking at fish sedimentary DNA. So ideally, fish DNA that's preserved in lake sediments. The thing that I find most compelling, or at least the most exciting, is the possibility to just reconstruct biodiversity over time. And that's definitely what kind of drew me to this program, drew me to Maine-eDNA, because that was one of the things they wanted to do with fish, or with alewife specifically. And it's been done for other species at different points in time, and it hasn't been done for fish, at least not as reliably. And it also excites me that that's an unknown to be explored. So sedimentary DNA, or sedDNA, is essentially a branch of environmental DNA. So DNA is shed from an organism into the environment. Often it ends up in water, and that's what a lot of environmental DNA samples. But over time, it settles into the bottom of lakes, oceans, reservoirs, and just becomes sedimentary environmental DNA. So I'd say one of the main general challenges for sedDNA is inhibition. So there being tannic or humic acids or other organic products just from trees, really anything that also just ends up in the bottom of those lakes. It interferes with the amplification of your target DNA. There's not that much DNA. And so, I've been using 40 times the amount of sediment to detect fish as one would need for plants or bacteria. All that really starts with, there's plenty of other sedDNA studies out there focusing on other species. So, the first thing we did was let's try what they're doing. And for a lot of times, that's kind of just using a kit that you can just buy to extract DNA from soils and just follow the manufacturer's instructions. When that doesn't work, then it's kind of just changing one thing at a time and seeing if you get the result you want. And so kind of the first thing I started doing was just increasing the amount of sediment used, going from a quarter gram to a half gram to a full gram, and eventually going all the way up to 10 grams of sediment. And then when that wasn't really working, then it's, all right, is there some way to concentrate the DNA after you extract it? And so it ends up just being a lot of tests, working with both increasing the volume of sediment, then both doing that while simultaneously concentrating it. It ends up being a kind of two-step extraction method where both you're concentrating the DNA firsthand and then you're extracting it with one of those commercial kits. And so for surface sediment in particular, one of my projects right now is trying to figure out how long that fish DNA lasts in the sediment, especially once you remove that point source. And, fisheries and wildlife and marine resources, they're particularly interested in that because knowledge of how long the DNA signal lasts could help inform them of look, we detected a Atlantic salmon. We haven't seen one in the system before, but, maybe that means that it was here within the last 4 months. And so next year, let's go ahead and adjust our sampling period to try and net this area within that time frame. A lot of different lake associations here in Maine are particularly interested in, were alewife in this lake 200 years ago prior to the onset of dams and other things like that. So I think being able to reliably go back in time and say yes or no, a certain species was there, was not there, is the most direct application and one thing that kind of excites me the most. And that's something that works for other species, and I feel like we're getting there for fish.